My name is Sarah Majin, and I'm a graduate student of Biomedical Engineering at the University of New Haven. And I'm here to represent my team members and the BMDI lab to present our research on dose-dependent cytotoxic and proliferative effects in mammalian fibroblast cells exhibited by liposome nanocarrier of signaling molecule. We took an interest in nitric oxide because it's a bioregulatory molecule that has diverse effects on the human body, such as hormone secretion, neuron transmission. But due to the short half-life of nitric oxide, it encouraged us to encapsulate it in a carrier before introducing it to the human body. And this can be used for various biomedical and clinical applications. On the image on my right, this is just an overview of the background of our study. We took a nitric oxide carrier, which is the liposomes. We inserted in the liposomes, and we studied the effect of the nitric oxide on fibroblast cells. And because fibroblast cells are crucial cells in connective tissues, and they're responsible for producing the ECM components. Also, they play an important role in wound healing and tissue repair. We had so many purposes during this research, but the main one was to investigate the role of nitric oxide in cellular processes, including apoptosis and proliferation. Some of the methods we used were thin film hydration methods to produce our liposomes using dried phospholipids, and we also used transmission electron microscopy to quantify the liposomes. Another method was to culture our fibroblasts and at different time frames, 24, 48, and 72 hours, and at different concentrations. Using the DSPC phospholipids, we were able to produce liposomes, and with our TEM, the transmission electron microscopy, we were able to quantify liposomes and produce about 40 to 100 nanometers of liposomes in size range. Although we also produced like smaller liposomes, which are shown in the white arrow, and the yellow arrow are the range of our liposomes produced. Apart from the size of the liposomes, we're able to get a zeta potential of minus 29.45 plus minus 0.07 tolerance millivolts and a PDI of 0.47. Using the nitric oxide analyzer, we were able to measure the nitric oxide release from 100 microliters on purified lipo-NO in PBS. And at different pH, at 3 and at 5, we carried out this experiment twice, and we were able to see the difference, the effect of pH in the release of nitric oxide. We're able to also see the effect of release in nitric oxide at a pH of 7.34, carrying out 3 experiments as previously. Also, we incorporated 1 ml of 1 molar HCl acid, and we saw an increased NO release after incorporating this acid. There was a huge spike in the NO release. So we also studied the interaction of the nitric oxide with the fibroblast cells to show the proliferative and cytotoxic effects at various concentrations. And at ratio 1:4 and 1:2 showed similar trend. But now, in addition to the 1:4 and 1:2, they both supported cell growth at 72 hours of incubation, unlike the 1:1. It also noted that the cells in both 1:2 and 1:1 media were characteristically defined by high amounts of granular cell membrane-bound and cytoplasmic structures. We also tested to check how the liposomes are spread out in the fibroblast by adding CFDA dye to the liposomes. In conclusion, the impact of our liposome nitric oxide conjugates, depending on the dosage and both the promotion and inhibition of fibroblast cells proliferation, it demonstrated a successful delivery of nitric oxide to the cells. So we're able to successfully deliver nitric oxide to the cells. And the random nature of this interaction, we know that in future, when it is harnessed using cell targeting ligands, has the potential to enable precise delivery of nitric oxide from lipo-NO within mixed cell populations. These are references, and all thanks to our professor, Dr. Keja Amoako, and all the students in the BMDI lab that contributed to this research. Thank you so much for listening.